The electron transport chain, ETC, is a critical component of cellular respiration, primarily occurring in the inner mitochondrial membrane of eukaryotes. Its main function is to generate a proton gradient, which is then used by ATP synthase to produce the vast majority of ATP, adenosine triphosphate, the cell's energy currency. This entire process is known as oxidative phosphorylation. Here's a breakdown of the key steps and components. 1. Delivery of electrons by NADH and FAD. 2. NADH and FAD2 are high-energy electron carriers produced during earlier stages of cellular respiration, glycolysis and the Krebs cycle. They deliver their electrons to the ETC, becoming oxidized back to NAD+ and FAD, which can then be reused in other metabolic pathways. The ETC consists of a series of 4 major protein complexes embedded in the inner mitochondrial membrane, along with 2 mobile electron carriers. Complex 1, NADH ubiquinone oxidoreductase, or NADH dehydrogenase. NADH donates 2 electrons to complex 1. These electrons are passed through flavin mononucleotide, FMN, and iron-sulfur, Fe-S clusters. As electrons move through complex I, it pumps 4 protons, H+, from the mitochondrial matrix into the intermembrane space, contributing to the proton gradient. The electrons are then passed to ubiquinone, Q, a lipid-soluble mobile carrier. Complex 2, succinate ubiquinone oxidoreductase or succinate dehydrogenase. This complex receives electrons directly from FADH2, produced during the Krebs cycle, and bypasses complex 1. FADH2 donates its electrons to complex 2, which then passes them to ubiquinone, Q. Complex 2 does not pump protons across the membrane. This is why FADH2 yields less ATP than NADH. Ubiquinone, Q or coenzyme Q, a small, lipid-soluble molecule that acts as a mobile electron carrier. It collects electrons from both complex 1 and complex 2 and delivers them to complex 3. Complex 3, cytochrome BC1 complex or ubiquinol cytochrome C oxidoreductase, receives electrons from ubiquinol, the reduced form of ubiquinone. Electrons are passed through cytochrome B and Fe-S protein, Reski center, and cytochrome C1. Complex 3 pumps 4 protons, H+, across the membrane into the intermembrane space. It then passes the electrons to cytochrome C, another mobile electron carrier. Cytochrome C, a water-soluble protein that acts as a mobile electron carrier. It carries electrons from complex 3 to complex IV. Unlike ubiquinone, cytochrome C can only accept 1 electron at a time. Complex IV, cytochrome C oxidase, receives electrons from cytochrome C. Electrons are passed through cytochrome A and cytochrome A3, which contain heme groups and copper ions. This is the final step where molecular oxygen, O2, acts as the terminal electron acceptor. Oxygen combines with the electrons and protons, H+, to form water, H2O. This step is crucial for pulling electrons through the chain. Complex IV pumps 2 protons, H+, into the intermembrane space. 3. Proton gradient formation, chemiosmosis. The pumping of protons by complexes 1, 3, and IV creates a high concentration of protons in the intermembrane space. This creates an electrochemical proton gradient across the inner mitochondrial membrane, with a higher positive charge and lower pH in the intermembrane space compared to the mitochondrial matrix. This gradient represents stored potential energy. ATP synthesis, oxidative phosphorylation by ATP synthase. The stored potential energy in the proton gradient is harnessed by ATP synthase, complex V. ATP synthase is a large protein complex that acts as a molecular motor. Protons flow back down their concentration gradient from the intermembrane space to the mitochondrial matrix through a channel in ATP synthase. This flow of protons drives the rotation of parts of the ATP synthase enzyme, which in turn catalyzes the phosphorylation of ADP, adenosine diphosphate, and inorganic phosphate pi to form ATP. This process is called chemiosmosis.